When you eat healthy, plant-based foods like spinach, beets, or almonds, they travel down into the dark, fluid environment of your colon. As these plants break down, they release a tiny sour compound called oxalate. Our human cells lack the biological machinery to digest oxalate. To our bodies, this compound is entirely useless. It is raw, indigestible chemical waste that must be managed. Because we cannot use it, the large intestine functions as a massive biological interception zone, a physical filter designed to trap this waste before it can spread to the rest of the body. This gut barrier serves as our first, most critical line of defense, neutralizing the chemical load long before it reaches the bloodstream or internal organs. This logic map shows how that defense works. It relies on a specialized gut bacterium called Oxalobacter formigenes. While other food molecules bounce away, this microbe captures and absorbs only the jagged oxalate. Oxalate is its exclusive source of energy. Once inside, the bacterium deploys specific enzymes that act as molecular scissors, slicing the waste apart and dismantling it into harmless gas and acid that safely dissipate. This primary consumer gets help from a backup crew. Other generalist bacteria in the gut support the process by digesting plant fibers and producing nutritious juices called short-chain fatty acids. These fatty acids act as a direct fuel source for your intestinal wall. They keep the cells healthy and tightly sealed together, locking the remaining chemical waste securely inside the interception zone. The specialized bacteria even send chemical signals directly to the gut wall. This communication activates microscopic pumps on the tissue, which pulls stray oxalate out of your bloodstream and dump it back into the colon for destruction. Managing this constant oxalate load depends on outsourcing our internal filtration to this cooperative yet fragile microbial workforce. That fragile microbial workforce is easily destroyed. A single course of common clinical antibiotics can cause instant chemical disruption, wiping out the protective bacterial colonies living along the intestinal lining. This creates a state of gut dysbiosis, an ecological void where the specialized cleanup enzymes simply no longer exist. Without these active consumers to eat the waste, the unintercepted oxalate rapidly pools into high concentrations within the unprotected colon. Deprived of their protective fatty acids, the tight seals of the gut barrier begin to fail. The chemical waste passively leaks straight through the biological tissue. This flow diagram tracks the escaped waste as it crosses the porous boundary of the gut and floods into the body's downstream transport pathway, the human bloodstream. The saturated blood travels relentlessly downward, carrying the toxic payload toward the body's final filtration checkpoint, the kidneys. What began as a microscopic loss of bacteria in the gut has now triggered a high-volume chemical threat to the major downstream organs. Inside the delicate cleaning tubes of the kidney, the renal tubules, the system becomes overloaded. This condition, known as hyperoxaluria, creates a dangerous chemical environment. Within these tubes, the highly concentrated oxalate aggressively binds with naturally occurring calcium, locking together into sharp, rigid mineral crystals. These heavy crystals drift through the liquid until they snag on tiny, rough patches of calcium lining the inner kidney walls. These patches act as anchors. Once attached, the crystals rapidly accumulate, stacking layer upon layer of hard minerals until they grow into massive, tissue-damaging structures that block the flow of urine. These hard, painful kidney stones represent the final physical consequence of a defense system that failed much further upstream in the gut. Because the root cause is ecological, biomedical science is shifting its focus toward repairing the upstream biological ecosystem rather than just treating the downstream damage. This strategy involves engineered bacteria, microbes modified to dismantle oxalate on command. In the presence of a specific seaweed sugar, the bacterium activates its internal enzymes and begins neutralizing the waste. To ensure these microbes survive, they are delivered as symbiotics, a package containing the live bacteria fed by the exact plant fibers they need to thrive. Once attached to the gut wall, they begin clearing the environment of jagged oxalate particles. Restoring the body's internal filtration requires active management of the upstream microbiome. When the interception zone is restored, we see a harmonious connection. The waste is neutralized in the intestine, keeping the bloodstream clear and leaving the kidneys free to function without injury. Protecting the microscopic life inside our colon is a primary way to secure the health of our major physical organs. From the intestines to the kidneys, the body functions as a continuous chain reaction, where the most effective protection begins upstream.